Assalamualaikum and good day to everyone, especially to our beloved instructor, Ma'am Sittie Jamelah Gumal. I am Roland S. Paulican and I am here to discuss to you the Vesicle Transport. I hope you will enjoy listening and happy learning! Vesicular transport is the predominant mechanism for exchange of proteins and lipids between membrane-bound organelles in eukaryotic cells. Golgi-derived copy-coated vesicles are involved in several vesicular transport steps, including bidirectional transport within the Golgi and recycling to the endoplasmic reticulum. In addition to protein processing, the endoplasmic reticulum and Golgi also take care of some types of protein transport. Vesicle or membrane-bound bubbles essentially pinch off from the Golgi and other membranous organelles, carrying with them whatever soluble molecules were inside the fluid that was enclosed as well as any molecules embedded in that section of membrane. These vesicles then catch a ride on a molecular motor such as kinesin or myosin and travel along the cytoskeleton until they dock at the appropriate destination and fuse with the target membrane or organelle. In general, vesicle move from the to the cis Golgi, from the cis to the medial Golgi, from the medial to the trans Golgi, and from the trans Golgi to the plasma membrane or other compartments. Although most movement is in this direction, there are also vesicles that move back from the Golgi to the carrying proteins that were supposed to stay in the and were accidentally scooped up within a vesicle. Vesicles bud from the endoplasmic reticulum and merge to form ergic, which matures into the cis Golgi, then the medial Golgi, and finally the trans Golgi. Vesicles may also bud from any of these other compartments to other organelles or to the plasma membrane. The formation of vesicles is dependent on coat proteins that will, under proper conditions, self-assemble into spherical cages. When associated with transmembrane proteins, they can pull the attached membrane along into a spherical shape also. The major types of coat proteins used in vesicle formation are COP2, COP1, and clathrin. COP2 coat proteins form the vesicles that move from to Golgi. COP1 coat proteins are used between parts of the Golgi apparatus as well as to form vesicles going from the Golgi back to the Finally, clathrin is used to form vesicles leaving the Golgi for the plasma membrane as well as for vesicles formed from the plasma membrane for endocytosis. The ARF1 or SAR1P is used to recruit adapter proteins that bind to the tail end of membrane-bound receptor proteins. The business end of these receptors bind to cargo molecules that need to be packaged into the vesicle. The adapter proteins act as the link between the membrane through the receptors and the coat proteins. Clathrin is the best described of the three, and the vesicular coats are made from arrangements of clathrin triskelions, from Greek meaning three-legged. Each triskelion is composed of three heavy chains joined together at the C-terminus and three light chains, one associated with one heavy chain. The heavy chains of different triskelions interact along the length of their heavy chain legs to create a very sturdy construct. The light chains are unnecessary for vesicle formation and are thought to help prevent accidental interactions of clathrin molecules in the cytoplasm. In the figure, letter A, clathrin binds to adapter proteins which are bound to transmembrane cargo receptors, linking the membrane with the clathrin. Letter B, a single clathrin triskelion is composed of three heavy chains and are three light chains. Letter C, the triskelions self-assemble into a roughly spherical construct without the need for any additional energy or enzymes. All three types of vesicle coat proteins have the ability to spontaneously associate into a spherical construct, but only COP1 and COP2 coated vesicles also spontaneously pinch off the membrane to release the vesicle from its originating membrane. Clathrin-coated vesicles require an external mechanism to release the vesicle. Once the vesicle has almost completed, there is still a small stalk of neck of membrane that connects the vesicle to the membrane. Around this stalk, dynamic GTP molecules aggregate in a ring or spiral construction. Dynamine molecules are globular GTPases that contract upon hydrolysis of GTP. When they associate around the vesicle stalk, each dynamine protein contracts, with a combined effect of constricting the stalk enough that the membrane pinches together, sealing off and releasing the vesicle from the originating membrane. The figure shows dynamine monomers, each of which is a GTPase, polymerizes around the neck of the vesicle when the GTP is hydrolyzed. The dynamine noose tightens and pinches off the vesicle. Glycerophospholipids are made primarily in the endoplasmic reticulum, although the ER also makes the ceramide precursors for sphingolipids. The sphingolipids are made only in the Golgi. The coat proteins come off shortly after vesicular release. For clathrin, the process involves HSC70 and ATPase. However, for COP1 or COP2 coated vesicles, hydrolysis of the GTP on ARF appears to weaken the coat protein affinity for the abductors and initiates uncoating. The GTPase activator is ARF-GAP and is an integral part of the COP1 coat. The vesicles carry two categories of cargo, soluble proteins and transmembrane proteins. Of the soluble proteins, some are taken up in the vesicle by virtue of being bound to the receptor. Other proteins just happen to be in the vicinity and are scooped up as the vesicle forms. Finally, there is the question of targeting the vesicles. The vesicles are much less useful if they are tossed on a molecular freight train and dropped off at random. Therefore, there is a docking mechanism that requires a matching of the V-snare protein on the vesicle's cytoplasmic surface and the T-snare on the cytoplasmic surface of the target membrane. Fusion of the vesicle to the membrane only proceeds if there is a match. Otherwise, the vesicle cannot fuse and will attach to another molecular motor to head to another, hopefully correct destination. This process is aided by tethering proteins which initially make contact with an incoming vesicle and draw it close enough to the target to test for snare protein interaction. And that is all for today. I hope I have imparted enough knowledge and information about the vesicle transport. Thank you for listening, and God bless us all always.